Zagotenemab is an anti-tau antibody, a humanized antibody against tau, but in particular, it is highly specific to aggregated forms of tau. It does this by being conformationally dependent in its binding. It binds both to the microtubule binding region in the middle of the tau molecule, but also to the N-terminus, requiring a conformational setup in order to bind properly. It's believed that the aggregated tau filaments are the type that are seated between cells. So we know that tau forms inside of the cells, but we also believe at least one of the mechanisms by which tau spreads is extracellularly. And as a large molecule, zagotenemab, which can't get inside the neurons, is targeted to those extracellular tau aggregate seeds, with the hope to slow down the spread of that mechanism of spread throughout the brain of tau pathology. So in PERISCOPE-ALZ, which is the name of the phase 2 study, it was designed to be a proof of concept study for efficacy, to demonstrate by both biomarker and clinical endpoints whether or not zagotenemab could slow the spread of tau and, as a primary objective, slow the clinical progression of the disease. So the study, PERISCOPE-ALZ, was set up into 3 study arms, all randomized equally, with 2 drug doses compared to placebo, a high dose, a low dose compared to placebo. And it was a study that was for 2 years in length with 104 weeks of observation. The primary endpoint measure was a composite cognitive and functional measure called the Integrated Alzheimer's Disease Rating Scale, which is a composite of both the ADAS-Cog-13 as well as the Instrumental Activities of Daily Living Scale as a summed score. Unfortunately, at the end of the trial, after 104 weeks of observation in these 3 study arms, there was no evidence that zagotenemab had a meaningful reduction in clinical progression on that primary endpoint measure, the iADRS. In addition, there was no evidence that it had any slowing of progression effect compared to placebo on the secondary endpoint measures, which included various items such as the ADAS and the IADL independently, as well as the CDR sum of boxes. So that if you also look at the biomarkers, then you say, okay, well, maybe the change wasn't sufficient. The study may have been relatively small as a proof of concept study that was designed to look for large effect sizes. But perhaps there were some underlying biomarker changes that could suggest that you were slowing that seeding of spread. We looked at both fluorotau PET, so tau PET, as our primary evidence of a spread of aggregated tau, neurofibrillary tangles across the brain. We also looked at neurofilament light in the plasma, as well as plasma tau, and volumetric measures on MRI. And in none of those measures, unfortunately, we did not discover any evidence that there was slowing or differences compared to placebo in any of those biomarkers that would suggest a disease-modifying effect of the zagotenemab molecule. Yeah, I think what's important to understand is, what this study tells us is that for one reason or another, zagotenemab did not impact clinical efficacy. It does not invalidate the tau hypothesis. It does not necessarily invalidate the hypothesis of spread of tau from one cell to another extracellularly. But we did not show evidence of that based on the sensitivity of the measures we're using, the length of trial, the impact of the drug. But I think to some degree, I think this does speak to the challenge of using large molecules for targeting tau and reducing tau spread, as tau is predominantly a pathology that starts intracellularly inside the neurons, and large molecules, we not only have a difficulty getting them into the brain, but we have some difficulty also, but once they're in the brain at a small proportion of our antibodies get into the brain, but those that don't get into the to the neurons and into that intracellular space that we know. So perhaps this study, as several other large molecules, anti-tau antibodies have now failed in phase 2 in the field. This is another one of those that has demonstrated how challenging this extracellular tau spread target may be. It may very well be that extracellular tau spread is still a viable target, perhaps by using different mechanisms, binding in different regions. But I think it also emphasizes that efforts to look towards small molecule therapies that can better penetrate and get to the targeted pathology are going to be important moving forward.